Yeah.
Hey, hi, Professor Lee and Johnson. Nice to meet you online.
No, thank you all for taking time off 除夕夜 just to make this call.
Yes, yes. I mean, thanks for organizing this. I think we are keen, so we want to learn more about this. Maybe I will before Johnson share. I
Mm.
so
Let me just stop
I
the sharing first, huh? 1 minute. Yeah. Okay, please go ahead. Thank you.
yeah. So today I'm actually more to actually wanting um, to actually hear more of the clinical side because this investment will also involve our group of doctors. We have some cardiothoracic surgeons that we actually form, and actually we they are also part of our shareholding group. So we would like to actually be able to explain to them. I would like to, I have to explain it clinically so that they
Yeah.
can actually have interest and then we can move on on this platform. So today actually that's my other than the other explanation, I would actually prefer this to be a bit more clinical so
Yep.
that I can actually explain to the doctors. Interestingly, I tried to read up quite a bit. I am still not very clear. I apologize, Professor Lee. I think this is really not my forte.
That's perfect. No, in fact, right, in the YouTube clip I sent yourself of a
Mm,
Saturday call a while ago, there was I think Professor Lee started with a 3D model of how exactly the AAV
mm.
gene therapy was delivered. The delivery
Yes.
mechanism is dramatically different from all the peer group in the com sheet, which you're very familiar about. And today is
Mm.
more of an interactive Q&A, right, where I can tell
Yes.
you that I've collated a bunch of very difficult questions from Obimed, from Boyu, even from ourselves, right?
Mm.
And then I'm going to ask those, repeat those questions. They've already received it before, right? Let them answer it. And then if you
Mm.
have further questions, right, let's drill them hard on this because because what we're doing here, right, is we are partners of the firm, right, of
Mm.
Medera. They're going to
Yes.
face this at the FDA shortly in April and May, right? That's
Right.
the litmus test. Okay. So Professor Hajar, who's actually the CMO and being the closest to the PIs, etc., especially regulatory, he's on, as well as Dr. Wang, right? But before I get started, right, I maybe I'll just say a few words. I just want to introduce the investor group here. My apologies. I wish to have done a call with each one of you separately, but of course, because it's CNY and we're about to go into a shutdown for a week, the
Mm.
the problem is the US side doesn't celebrate CNY. It's our Christmas. It's not theirs. So
Mm.
the US investors are actually moving ahead. And I was kind of worried that if one of those major players, right, they just pump the table, right, and actually the round is closed. That's a, a significant probability event. That was why I
Mm.
asked the professors to come online and just give us as much of a download so that we can sit on the data through Christmas, right? And all these are recorded. I'll put this on my private YouTube for you guys to review that. Okay. But
Mm.
yeah, so actually I had an agenda going in, but before that, I would just want to introduce the two professors and their team, right? So I would just repeat this part, which really impressed us and Met a lot, is that Madeira did not receive any institutional funding for the entire 10 years of their survival, right? Or their origination. One key reason is that they received 130 million US dollars of NIH funding grant across three founders. That number must have been is absolutely the highest that we have seen within Optimus and IQVIA. But maybe for Albimed, they mentioned that only Moderna has received a higher number. And why is this NIH funding the golden weight, right? It has its weight in gold, is because the grants are given from NIH, DoD, this kind of purely on scientific merit, not on potential commercial scale or VC funding, none of that, right? Non-dilutive funding. And in fact, when we arrived in Singapore, I think around 15th of November last year, where we met Dr. Lim, who is the bridge to get us all together, That
Mm.
day, Ronald received 15 million Sing dollars from, I think, A Star plus National Heart Foundation, right? So these are very heavyweight funding, which means that the Singapore MOH is already pushing this forward. In fact, I will let Ronald just introduce this part and the two professors' background, and then tell us more about the NIH funding. Yeah, please, Ronald.
Yeah, thank you, Johnson. It's nice to meet all of you. So I'll quickly introduce the group and also myself, and then I'll pass the baton to Roger and Fubao to do their introductions as well. So Medera was founded about 10 years ago in 2014 as an academic spin out of Johns Hopkins University and Harvard, where our original technologies were first developed primarily by Roger, myself, and also our colleague Kevin Coster, who is not on the call today. And the company, although it's been around for 10 years, but the technology, as I briefly hinted, so it's really something that the three of us have been working on for over a period of 25 years for myself, and Roger has been my senior colleague the whole time, for him 30 years. And we all came from the same pedigree of Johns Hopkins Biomedical Engineering and Cardiology. So we have been very privileged to have become involved in the area since the very beginning, when it was nothing but just the concept. So basically, Medera was founded, and before this. We have been, Roger and I have been basically just licensing out our technology for other companies to develop. But then 10 years ago, we decided to found our own company to really try to develop the technologies and make decisions as we see fit. In a way, Medera is really focusing on the commercialization aspect, or pretty much like the final step
Mm.
of what we think has been a 30-year journey, and we're going to tell you more about it. What we believe is really unique to Medera is that I'll very quickly go over, and there are two aspects. So one is that we have this proprietary delivery method that we call intracoronary delivery protocol, by which we can directly inject a drug into the heart. And it serves two main purposes. One is that it basically eliminates all side effects. So Roger is going to tell you about the clinical data as we have observed and also independently confirmed by Bayer as well. So I'll have time to go over the relationships and the history among these three several companies and the several generations of gene therapy that we have personally witnessed. And the other unique aspect is that we have the so-called human mini heart technology platform. Uh, and this is really our discovery engine, which allows us to discover, validate, and also even optimize dosages before we put anything in human. And as Johnson mentioned, so before we founded the company, we managed to secure basically $150 million for the three founders, 130 million between Roger and myself, 100 million for Roger more specifically, 30 million by us, and 20 million from the remaining third co-founder. And in Singapore, we had just received this national level kind of like grant from A Star for supporting the collaboration that we are having with National Heart Center. And more specifically, this is about using or building a Singaporean or Southeast Asian mini heart library. So meaning Chinese, Malaysian, Indonesian, and Indian to focus on heart failure in Asian. So that's for Medera, and for myself. I was one of the co-founders. So Roger and I and Kevin Coster founded the company. Within the group, I'm functioning as the CEO. And before this, I was an academic. I was a professor for over 20 years, and I started some of the work that we're about to present to you when I was an assistant professor at Johns Hopkins, and I went through the rank at several other U.S. and institutions. And about 7 years ago, I decided to be full-time at Medera, trying to, as I said, focus on commercialization and also to try to push the final step. And we'll be very excited and glad to tell you more specifically about some of our ongoing clinical trials and the corporate plans that we have for Medera. Thank you. Roger?
Hi, thanks, Ron. So I'm Roger Hajar, and I've been working with Ron for over 25 years now. I'm a clinical cardiologist, heart failure transplant specialist. So I focus on congestive heart failure as my clinical career. Um, I've been working in the field of cardiac gene therapy now for close to 30 years, and really focusing on the clinical aspect of bringing gene therapy to patients for the last 20 years or so. Currently, I'm the head of the Gene and Cell Therapy Institute at Mass General Brigham and Harvard Medical School. So I oversee a large group of people that focus on gene and cell therapy. So I'm well aware of all the nuances of gene therapy in many diseases, including cardiovascular. And within Medera, I work as the chief medical officer overseeing the clinical trials. And as Ron mentioned, it's been really uh, kind of a roller coaster journey, but we feel that we've learned so much over the years with the first, second, and now the third generation of gene therapy that's focusing on cardiac diseases. And as I will mention, we're very excited about the data that's coming through with the heart failure preserved ejection fraction, a big, big problem with big unmet need that has a lot of patients that are not being served by the current medications. So we're very excited about the data, and we feel that this is gonna take Medera to the next step in terms of being a viable commercial entity in a space of gene therapy for a large population, not just rare diseases or ultra rare diseases with a very small population. And to do that, we're very fortunate to have Fubao, who's our regulatory head, who's really guiding us through the regulatory path at the FDA. So maybe Fubao, you want to give a quick intro.
Thanks, Roger. Hello, everyone. I'm Fubao Wang, and I joined Medera in August 2025. And so I've been in the industry for 28 years. Now it's on the way 29 years. I've been trained as a molecular biologist and started my industrial career back in 1997 at Merck, and then stayed there for 17 years and worked on the vaccine and the biologics product development and later regulatory on the global market. And then I worked on another pharmaceutical company, Sanofi, and in a regulatory role. And then I switched over to the biotech companies for Sarepta Biotech, AskBio, and the Prime Medicine, and now it's Medera. My association with the regulatory has been 14 years, and in particular with AAV-based gene therapy products since my time at the Sarepta, it's been 80 years. And I was attracted by the really the solid science and with prominent scientists like Roger and Ron in the field. You heard from them the 20, 30 years working in the field, really have deep science in this field. And also the disease target in heart failure, which has such a profound impact on the general population. I think if and this approach, naive-based gene therapy, and the once it's developed, approved, will have a very profound impact on the public health of the heart failure patients. And that's, to me, as a regulatory person, and it's going to be very excited to be part of this team. And we work hard really trying to move this program through the clinical stages. What we hope is working with FDA and other regulatory agencies and trying to bring this product, go through the clinical phase and bring to the patients ASAP. So I'm happy really trying to discuss and answer any questions in that regard. And all in all, I just really want to share my excitement with the program and the data we have. And I stop here, maybe come back to Ron.
Yeah. No, actually, let me just jump in, right? I'm going to point out Dr. Wang is actually the key point person facing the FDA pretty shortly, right? Even right now with your RMAT, we will come to that, right? Which is really the breakthrough therapy designation for in an oncology setting, right? But RMAT is for regenerative medicine. But I just want to quickly give 1 minute, right, to Dr. Michael Lim, who is really the bridge between all of us here today. He's the actually founding editor of the Asia Heart Journal, right? Also, David Howe, my buddy at IQVIA, who is actually the lead investor in this ticket, right? Maybe just 1 minute of brief intro, because I've mentioned you guys many times to your group of investors. Please, Dr. Lim.
Hi, I think know some of these people. I think Yongping we've met before.
Yes.
Yeah.
Hi, Michael. How are
Hi,
you?
hi.
Been a
I'm good,
long
I'm
time.
Yeah, so basically, I'm currently part of a global conglomerate. I'm also same destination like Roger. I'm the CMO. And basically, what I do is that I interact quite a bit with all the biotech companies, the medical devices companies, and then I evaluate them for the group, and I'll see where, can we work with them to grow the technology or grow the space. So that's where I am now. We're part of but I'm just looking after the healthcare sector. Yeah. I'm a cardiologist by training. I'm quite unusual because I train in cardiology. I went to US and I did a fellowship in interventional radiology, so very unusual. And also I have a postgrad in sports medicine as well, plus another two masters in internal medicine, one in Singapore and one in Royal College of Physicians in UK. So I pretty much, you can pretty much discuss most things in medicine with me, and I wouldn't say I know everything, but I would know something about most things. Yeah. Yeah. Thanks, Johnson.
Thank you, David.
Yeah. Michael is also a previous member of the parliament that I think he didn't mention. So he's actually very well connected.
Yes. Optimus has invited Dr. Lim on as our SAB and of course Medera as well. So we're very glad to have that. So David from IQVIA, IQVIA China Fund, please.
Yes. Hi, Joseph. Hi, gentlemen. Yeah, I'm David. And our fund is actually backed by IQVIA, the world's largest CRO. So basically, the largest contract service provider for clinical trials, and also the largest healthcare consulting and data firm. So our focus is actually to invest in drugs across various sectors. And cardiovascular is actually one area that we are keenly focusing on. So Medera is a project that we've been following over the past, I think, over 2 years now. And we do like the project a lot. So for this round, we're not only the lead investor, but our headquarters back in the US are also entered into collaborations with a company on two fronts. One is providing CRO services for future clinical trials, both in the US and also in China. And also we've engaged in BD collaborations with the company, especially for the moment on Mini Hearts, the novel heart platform, and also in the future for licensing out opportunities once we enter into a mature stage. So we're currently based in Shanghai, and we do have a global reach for projects from all over the world. So Johnson, I will do a brief intro for now, and I will give it back to you.
Yeah. Okay, great. So, Professor, because actually most of them have received the detailed clip that we did last Saturday, right, with another group of investors. But I'm going to just focus on the toughest part of the question, AAV gene therapy, right? That's the label you're going to come with, and that's actually comes with a negative connotation because of Sarepta and Rocket and guys, right? So maybe you start us off, right, by showing us your proprietary delivery mechanism, right, the clip, and then tell us about your advantages of endorsing as well as Novo Heart, right? I think that should set the stage for Dr. Peng. Yeah, please.
Yeah, so let me share my screen. Can you all see my slides?
Yes.
Okay. So there are a few slides that I want to show all of you and should allow you to get a better idea. I mean, as I said before, and I'll show you this slide basically shows you, summarizes to you why we think Medera is unique, not only in cardiac gene therapy, but also gene therapy in general. So whenever people talk about AAV gene-based gene therapy, people usually are very concerned about side effects, life-threatening toxicities that are potentially associated with AAV. That's also is a result of the use of immunosuppression like steroid. Typically, most companies administer gene therapy drugs into the body by IV intravenous administration, and as such, the patients need to be hospitalized for about 7 to 10 days. Because of the vast amount that they are injecting into the body, the cost of good is also typically quite high, about $100,000 to $200,000 per patient or per dose. And as Roger mentioned, typically gene and cell therapies are focusing on rare diseases with very small patient populations, and everybody has been complaining that these drugs are very expensive, ranging anything from $1 to $4 million, such as the Sarepta AAV drug for Duchenne muscular dystrophy. For Medera, we're unique in several ways. So number one, Roger is going to go over more clinical data with you, is that we absolutely are not using any immunosuppressive therapy, and we have not seen any severe adverse events, side effects as a result of immunosuppression. We are seeing no T-cell responses, absolutely no liver damage as we monitor the liver enzymes during the course after infusion. So that really can be attributed to the unique intracoronary delivery methodology that I was kind of like briefly describing to you, and Roger will go over how exactly that is done and how we inject into the coronary artery. And by doing so, this is basically just an outpatient procedure that can be completed within half hour or so by any trained interventional cardiologist, as opposed to 7 to 10 days hospitalization, which add up the hospital bills, as you can imagine. So no side effects, no immunosuppression as an outpatient procedure. And also by injecting directly into the heart, the amount of viruses that we're injecting are basically 100-fold less than that of IV infusion. As you can imagine, the cost of good is also proportionally 100-fold less. And in the US, we are really talking about $1,000, $1,000 US per patient per dose. So as you can imagine, this is highly scalable. And also, we strongly believe that when the time comes for us to commercialize, our pricing can be very, very highly competitive. And also, we are talking about a common disease, heart failure, which afflicts 64 million patients worldwide. And in parallel, we also have a rare disease approach, and this is for Duchenne muscular dystrophy cardiomyopathy. Our technology is also first in human. And this is, as Johnson might have told you before, and this is basically our safety net. And for now, our focus really is on half-path, heart failure with preserved ejection fraction. And in a minute, I'll tell you a little bit more. And Roger and I recently published this article about gene therapy, AAV-based gene therapy, and we talk about the economics and also the importance of the safety aspects before we even think about efficacy. Happy to share this article with you. And this slide, I mean, as I mentioned before, we are taking this common disease approach, focusing on heart failure, which is for 64 million patients worldwide, about 14 million patients just in the US and EU alone. And there are two major forms, each accounting for about 50% or half of the entire heart failure population. This is really a global pandemic. So, and so far, we have injected 21 patients. And as I mentioned before, very importantly, the safety signals have been excellent. No liver enzyme abnormalities, no T cell responses, no immunosuppression therapy, absolutely zero steroid, and not even a single severe adverse event has been observed, and that's been documented at the level of the DSMB data safety monitoring committee at the FDA, and also as monitored by the FDA. And for now, our marquee program really is half-path heart failure with preserved ejection fraction, which accounts for 50% or half of the entire heart failure patient population. And for this, we are very excited about it because this is first in human disease-modifying drug, gene therapy. And with this, we are really, as to the best of our knowledge, we are the only company working on this area. The next slide, I'll quickly introduce to you first generation technology developed by Celadon and second generation by AskBio. And for Celadon, they started about 20 years ago, the first generation technology. They really were just focusing on HFrEF, remaining 50% of the entire heart failure population. And for us, our technology covers both ischemic as well as non-ischemic populations of the entire HFrEF populations as well. But strategically, we are really focusing on just the ischemic patient population, which is 50% or half of the HFrEF population, and therefore 25% of the entire heart failure population. So if there is time, we'll tell you, Roger will share with you some clinical data as well. But as I said, for now, our focus really is on HFpath. And AskBio Bayer, second generation AAV-based cardiac gene therapy, they are really looking at just non-ischemic population of the HFrEF patients only, so 25%. So as such, you can see that Medera's technology cover 75% or basically 100%, if you want to think about a non-ischemic population that we can target as well. So 75% of the entire 64 million patients. And this slide summarizes to you first generation AAV cardiac gene therapy. It started by Celadon about 25 years ago. Second generation Bayer, AskBio, cardiac gene therapy, and what we think Medera's technology is first generation. So of note, the Bayer's technology, they licensed from Rogers Laboratory about 15 years ago before we founded Medera. So we are very familiar and very, very excited to have seen the evolution of first, second, and third generation and how the technology has been changing. I'll very quickly summarize to you how we are similar and different. So all three companies, all three generations target the intracellular calcium handling pathway, focusing on the ultimate effective protein called SERCA2a, which is a calcium ATPase pump. And all three companies also using or have used the intracoronary delivery protocol, except the Celadon was using an O pump. We are now using an electrical pump to really control the infusion. AskBio is injecting manually. At the time when Celadon was doing it, sophisticated mini heart technology was not available. So they were basically jumping from animals all the way to human, as typically done by companies in the past, and also many companies, our current companies as well. So as I briefly mentioned, the mini heart technology is uniquely available to Medera, and also we happen to be the first cardiology company to have successfully secured FDA clearances and also our fast track designation by using the human mini heart technology, the data collected using the human mini heart technology platform. These are some of the technical differences, but I think they're extremely important. So the formulation that we use for our drug product is very different now. We have a new formulation that allows us to stabilize the AAV particle. And this is a very important difference. So back then, the state of the art, the manufacturing method was different for Celadon. They were getting as much as 70% what we call empty plasmids or empty capsids. So meaning that the AAV vehicles were empty, 70% of them without carrying the transgene, the calcium ATPase pump that we want to transduce, put into the patient hearts. So 30% for Bayer, you see that 1% for Medera. So this is a huge difference. 1% empty capsid as opposed to 70% empty capsid. And also the dosages that we are using are also very different. So back then, FDA capped. So Celadon wasn't only the first cardiac gene therapy, it was also one of the very first gene therapy companies. So back then, they were flying blind. They didn't know how much to inject into human, and also FDA capped the dosage that they were allowed to inject into human patients. So these days, companies like Sarepta, Rocket Pharma, they are typically using dosages that are easily 100 or even 200-fold higher than what Celadon was using. So we are also, we meaning Medera and Bayer, using higher dosages compared to Celadon. And I'll tell you how dramatically this affects or influences the efficiency in the heart. So 99% transduction efficiency as shown by human heart biopsy, as opposed to 0.5% back then. So, very low dosages that Celadon was able to use, a lot of empty capsid, which collectively explain why only 0.5% efficiency in the human heart back then, as opposed to 99% these days. So without getting into the details, so these are all given in our clinical protocol as approved by the FDA, allowed by the FDA. So patient selection, endpoints, and also they were not using neutralizing antibody screening. As a matter of fact, back then, AAV9 wasn't even discovered. So they were not aware of the issue of having to think about neutralizing antibody for the AAV serotype that they were using. And none of the companies saw any major or any severe adverse events.
Professor, sorry to interrupt, right? I've seen your pitch before. Exactly. I was looking for this slide and also the video clip about how the delivery is a lot more visual impact for the doctors. Please, thank you.
That'll be the next slide. So this basically summarizes to you the different dosages that the different companies like Celadon, Medera, Bayer, Rocket, and Sarepta is not shown here, but also very similar, like they were using some dosages somewhere here. So as I said before, Celadon was not allowed to use higher dosages as capped by the FDA. And also, if you consider 70% empty plasmid, so effectively they were injecting at this level, and as such, they were getting basically 0 or 0.5% efficiency. And when we go up by using higher dosages, we're able to get 100% transduction efficiency, as shown in human biopsy. So Medera, AskBio, and you can see that none of these companies observe any T cell responses except when Bayer AskBio went up to 1 times 10 to the 14 VG per patient. They started seeing some transient mild LFT and T cell responses. So, and for Rocket, they have reported patient death as a result of the triple immunosuppressive therapy. And for Sarepta, they saw liver damages when they injected such astronomical doses, or 100-fold dosages higher than what Medera and Bayer are using.
Just on
So
this slide, yeah, just on this
yeah,
slide, I just want to jump in, right? Because I keep telling the investors to take note of two numbers. One is N equals 10, 10 patients for our efficacy test in HFpEF, right? But over 21 patients in safety within our own trials, right? Well, therapeutic doses, right? But can you just explain how much can we read across from the AskBio doses, which because of the Novel Heart program, right, is exactly the same as ours? How much can we read across into their safety data, which is a lot more patients, and into their efficacy for HFrEF?
yeah, Bayer, I think they have so far looked at about 50 patients or so. Roger can confirm the number. So they,
Yeah, so Bayer, Aspire Bayer has dosed 110 patients, also with no immunosuppression, um, intracoronary, and at lower doses than Sarepta and Rocket and other companies, and they've seen no adverse events, just like us.
thank you, Roger. So this slide shows you the competitive landscape. So Bayer is here, Medera. So we have 3 ongoing clinical trials, all for cardiac gene therapies. Most other companies have 1 or 2, and preclinical. And for Rocket, so we were talking about the high dosages that they're using, and pretty much like everyone else is doing IV injection. And Bayer, as Roger mentioned, so intracoronary delivery protocol that allows them, they basically have validated what we've been saying is a lack of any side effects, and we don't need to do any immunosuppression. Sarepta is here, intravenous, also very high dosages. They reported patient death as a result of liver damage. And several companies, so looking at the same target effector. So this is a common pathway that many companies in the past and also current companies are trying to target. As I mentioned, so we're looking at HFpEF and HFrEF, and strategically focusing on an ischemic population of HFrEF, so thereby covering 75% of all the 64 million patients. And for Bayer, they're looking at just non-ischemic. So basically, you can kind of like imagine that we are not, really competing against each other. So this is the competitive landscape, and for now, this slide shows you our pipeline. So we are really focusing, laser focused on the 3 ongoing clinical trials, with HFpEF being our number 1 top priority asset. Duchenne muscular dystrophy cardiomyopathy, this is also first in human. This is our safety net, and we are running HFrEF in the background as such. But we also have a number of we have 4 other gene therapy preclinical assets with pulmonary hypertension, IND ready. We had the pre-IND meeting at the FDA, and working with IQVIA, we are trying to advance that to the clinical stage. And we have also several small molecule program as well. So Roger, maybe I'll let you take over and talk about the intracoronary delivery method, and also go over the clinical data.
Sure. Yeah, so this is a video that we produced a number of years ago to basically show the intracoronary delivery. And basically, it's a routine cardiac catheterization, which in the old days used to be done from the femoral approach, and now it's done from the arm, and from the radial artery. And the catheters are placed in the ostium of the left main and the right coronary. and are injecting downstream of those. So you can see here the green vectors that hold the therapeutic transgene, and these are the myocardial cells. They go into the cardiac cell, into the nucleus, and they produce message, mRNA, which then produces the protein that's deficient in heart failure. And then the protein induces functional improvements of the cardiac cell, resulting in an overall improvement in the contraction of the ventricle, both contraction and relaxation. So this procedure is been done very safely in the cath lab. It takes about half an hour to do the whole prep, and everything else is about 45 minutes. And the infusion is 10 minutes down each of the left main and the right coronary artery. Now, if the patient has bypass grafts, then that could be adjusted to cover as much myocardium based on where the flow of the bypass grafts are. But in our heart failure with preserved ejection fraction trial, we had two cohorts, one at low dose, 3E13 viral genome per patient, and then we have a higher dose cohort at 4.5E13 viral genome per patient. And this is sort of the usual patient population. So mainly EF is more than 50%, a higher propensity of females versus males in the HFpEF population, which is well known. These patients were on maximal therapeutics, all on diuretics. Some of them were on ACE inhibitors and ARN inhibitors, along with beta blockers, SGLT2 inhibitors, and MRA. Some of these patients were also on GLP-1 agonist for obesity related to HFpEF. But in general, a lot of comorbid illnesses in these patients. So this is a heat map where we look at different clinical parameters in HFpEF. New York Heart Association is the first one. The wedge pressure is another one, which is extremely important. 6-minute walk test, NT-proBNP, and KCCQ are other parameters. What we see here is that in the New York Heart Association, most patients improved from 3 to 2, and the one patient who was 2 to start with continued to be 2. And then we'll go into the mean wedge pressure next, because this is really where the important parameters come into play. That's where we do see the dramatic changes in the wedge pressures. And then most of the patients, as you can see on the right side, improved in terms of their KCCQ, which is a quality of life questionnaire. Slide. So this is really an important slide to show that the mean wedge pressure, both at rest and at peak exercise, decreased more than 30% in these patients at 1 year at the low dose. And this is equivalent to a 47% reduction in mortality risk. And this is really the largest improvement in wedge pressure versus other phase 1, 2, and HFpEF trial where wedge pressure was involved. And again, there was no immunogenicity or liver enzyme abnormalities or other adverse events. And when we compared the changes in New York Heart Associations, as you can see here in these patients versus external controls, which had similar characteristics of HFpEF, we see the vast improvement in our trial. Similarly, in 6-minute walk test versus other trials, and then mean changes in KCCQ at 12 months in our patients. So the wedge pressure was not a standalone improvement, but really was associated with improvements in other parameters. And the external controls, I think what's important is that when you look at other HFpEF trials, their parameters 70% of the time worsened were unchanged. So clearly, this is very different response in our gene therapy trials for HFpEF compared to other external controls. So just to give you an idea of the type of patient that we are dealing with, this is a patient who received a low dose, 63-year-old woman. And just look at the list of comorbid illnesses: asthma, dyslipidemia, hypertension, hypothyroidism, pulmonary hypertension, Raynaud's syndrome, scleroderma, and heart disease. But the one thing that really makes her go into congestive heart failure is her uh, preserved ejection fraction induced heart failure, so the stiffness, the diastolic dysfunction. And based on our disease modifying treatment, we're able to make this patient feel much better. So she went from class 3 to class 2. Her KCCQ improved tremendously. And you can see that her wedge pressure at 12 months at rest improved by 42% and at exercise by 55%. So clearly, she had a dramatic improvement in her functional parameters, and despite the fact that she has so many comorbid illnesses to begin with.
Professor, can you just take this chance, right, to talk about the other two metrics? I know that they start with a low base. Sorry, if you just go back one slide. Yeah, the NT-proBNP, right, etc. Yeah.
Yeah, so the KCCQ, which is a quality of life questionnaire, increased. So 0.5 is a significantly or clinically relevant improvement, showing disease modification, as you see with the wedge pressure. In terms of the NT-proBNP, usually these types of patients have varying NT-proBNP depending on the loading of the ventricle. In this case, the patient had a relatively low NT-proBNP, so a normal range of NT-proBNP, and they didn't seem to change much from that low range. So there was kind of a normal range for the NT-proBNP that did not change. But the most important parameter for us is really the wedge pressure, because wedge pressure really has a very specific HFpEF characterization and also has a direct influence on mortality.
Got it. I just want to remind investors, right? This slide that you're seeing is on the low dose, right? There are 5 such patients. Obviously, one of them, even on the low dose, which wasn't really supposed to show efficacy, almost reversed the entire HFpEF indication, right? Yeah. Now we take a look at the high dose, and then in the interest of time, right? Because we sort of have a hard stop at 11 for a different call. We have to hop on a different Zoom. I would like to actually quickly go to Dr. Wang's side about regulatory pathway. Yeah, and then leave Q&A questions for Dr. Peng, right? Please.
Sure.
So Ron, you're going to show the slides?
Uh,
Or should I just go over verbally?
Yeah, the
yeah,
high dose part, right? I think we still want to touch on the high dose part, and as well as what the PI from Duke is saying qualitatively.
so let me.
So you want to go through the high dose patients first, then go to the regulatory, right?
So in the high dose, maybe we can go to the next slide. Yeah, so basically, the high dose patients were all dosed right now. We have some data on the first 3, where at 6 months we see really a dramatic improvement in wedge pressure, similar to what we saw at 12 months in the low dose. So what we're seeing in these patients of the high dose is an acceleration of the improvement that we saw at the low dose, to such a degree that the principal investigator at Duke University, Murat Fudem, who's an expert in diastolic dysfunction, basically said that two of the three patients at the high dose at 6 months do not meet HFpEF criteria in terms of any evidence of diastolic dysfunction or signs of heart failure. So clearly, this is again directly from the PI who said this to us, in an email unprompted by us, just because he was couldn't believe how well these patients responded.
Professor Hajar, can you just help us out, right? If the high dose at 6 months is looking like that previous bar, where would at the 12-month point, where is projected to go to, as well as what does it mean in terms of death risk reduction?
Yeah, so I mean, at 6 months, they've already shown a 50% improvement or a projection to improve mortality by 50% already just at 6 months. And the hope is at 12 months, there will be a continued acceleration of the reversal. So even a further decrease in the wedge pressure, meaning that they will have demonstrated a tremendous reversal in mortality. And again, these are very different from what we see in other drugs that are used for HFpEF, such as the SGLT2 inhibitors and the Entrestos, here as shown here. So we see changes in Entresto. These are the responders at 11%, SGLT2 inhibitors at 15%. TX45 is really for pulmonary hypertension. But again, our low dose beats out all these changes in wedge pressure. So clearly, what we see here, both at rest and at peak exercise, are at the low dose are tremendous improvements in wedge pressure. And we're seeing even a bigger increase in the high dose. So we're quite excited about that.
No, this is perfect. Just for the generalists in the investor group, Entresto is the number one seller for Novartis, okay? And Farxiga is the number one seller for AstraZeneca, to give you a sense of how massive this drug is. And they are not selling their 20 billion sales combined, right? It's not for heart failure. They just got approved for heart failure 2 years ago, right? With very marginal impact. So it's worth looking at what their death risk reduction is in their trial. Yeah. Please. Thank you.
I would like to supplement by saying that keep in mind that our technology is one shot. So one shot, and then we measure the improvements at 6 months and after a year. So this is a lot more stable than conventional chronic pharmacotherapy. And maybe Roger, go over like some of the considerations for pharmacotherapy and the side effects that go with them.
Yeah, so I mean, I think with any drug that you give chronically over time, so whether it's Entresto or the SGLT2 inhibitors on a daily basis, you have specific side effects. So for Entresto, you have flushing, you have low blood pressures that can occur, or episodes of hypotension. With the SGLT2 inhibitors, you have a lot of abdominal side effects that have occurred in patients, sometimes not being able to tolerate it. So these are, once a day pills that you have to take over many, many years. And despite that, there's really not much of a disease modification as you go forward.
And before we go to Dr. Wang, right, I received a couple of questions on Novo Heart's contribution to how you guys find the dosing and why are we so different, right, from the other AAV gene therapies in dosing terms, right? So can you tell us how big an edge and advantage that is? Yeah, before I go to Dr. Wang on the regulatory timeline. Thank you.
Yeah, I mean, sorry, go ahead, Ron.
Go ahead, Roger, please.
Yeah, so the Novo Heart technology is really a human engineered tissue that gives us direct information about how human hearts will respond. So the dosing was based on a lot of animal studies, true, but also verified by engineered heart tissues. And now that the FDA and both the government are looking for alternative to animals, this has been really a good way to sort of bring that in. We're able to show that, as Ron is showing here, is that in these engineered tissue, the dosing was very much equivalent to what we had seen in humans that were showed an increase or an improvement in the dosing. Now, the difference between us and other cardiac gene therapy companies is the fact that we deliver by intracoronary means, so we can really reduce the amount of vector that other companies use. So our cost of goods are 100 times lower, and our safety profile is much, much better because of that.
And showing you here is the HFpEF sick heart that we co-developed with AstraZeneca. So we've been able to make normal as well as the HFpEF sick mini hearts. And combining this, the biological preparation with the hardware and software for automated screening, we're able to reproduce the disease phenotype. And this shows you that this is a normal phenotype, and with the disease sick heart phenotype, and by putting the exact same drug, gene therapy, AAV, circa vector, that the same batch that we are putting into human patients, we're able to reverse the phenotype. So this is really the key data that we are submitting to the FDA for getting our IND clearance and also fast track designation.
Can you just flash the Astra clip just briefly? Thank you.
Sorry. So are you still seeing my slide? Maybe we can forget about it. Somehow it's not working. So
Yeah, no problem. I can
share
answer
it.
separately. Cool.
Yeah.
So if you don't mind, right, in interest of time, I just want to quickly go to Dr. Wang and see if, first of all, I just want to share my slide. This is a specific question, okay? On the other regulatory AAV approved gene therapies. So look at the number of patients, right? Can you see that on the bottom right? This is AI driven, okay? So, Dr. Wang, I just want to ask a quick question, right? You were involved in let's say, the Sarepta ones in approval, which I think is a very knife-edge drug. The peak market cap of Sarepta was 15 billion US for an orphan drug. So when you got that through, right, on this list, on that, let's say the DMD line, right? What do you think about the regulatory pathway for us right now? Can you just focus on the timing of the RMAT, the significance of it, as well as in using this 10% data, right? When we go discuss with the FDA, is it a lot out of line with these guys who are going after orphan drug, right? And whether we might have a very good shot at getting a accelerated pathway, right? Please.
Yeah. Thank you, Johnson. And in the interest of time, and I will be really quick. Now, RMAT was submitted a few weeks ago, and we will in 60 days, we will know FDA's decisions. And if we get RMAT granted, that would increase our flexibility to interact with FDA. And we will get the guidance from FDA for the design of the next stage of clinical trials. And so what we are planning here is right now, so with the encouraging data that Roger just showed, we actively discussing and also seeking for advisory board opinion inputs, really try to see what's the best trial can look like. And what we are planning is to have a type B meeting in the second quarter, third quarter, to just propose a next stage clinical trial with the goals, and if everything works out, and we would have a randomized placebo-controlled double-blind trials, and for if the data looks really promising for accelerated approval, I think on the disease, there's a huge unmet medical needs, and it's our responsibility to bring this medicines to the patients ASAP. And so what in terms of the trial size, and certainly you saw the number that Johnson showed in the earlier slides, and we can also really try to go through statistical modeling, really trying to build the statistical powers into it for the heart failure. And for initial clinical accelerated approval, if FDA agrees, and we think we'll probably just propose, somewhere between 150 to 250 patients. And that right now is a little bit premature for us to say specific numbers. And we will in end of day to propose a statistically powered number and trial size and take that on to the FDA.
On this point, right, Professor Hajar, if you just jump in, right, last week when I asked you about what does the FDA consider primary, right, in whether accelerated approval, you mentioned safety, right? Can both of you give us a view on how important is safety in coming to this decision, as well as how it benchmark against the 6 which are already approved AAV gene therapies? Thank you.
Yeah, I mean, I think the safety is primordial in terms of the FDA's approach. They do tolerate some toxicity based on how rare the disease is and how big the unmet need is. But in our case, we're showing efficacy without any toxicity. So clearly, it's very different from many of these agents where you have to use heavy immunosuppression to basically get the effective dose. So I think from that perspective, it's extremely important.
And I want to quickly supplement that when you see an average change of 35%, and the reactive patients are even showing over 60% reduction in the wedge pressure, you don't really need to have like several hundred or several thousand patients in order to show the statistical significance. So at this point, obviously, we're not here. I mean, FDA is going to make the decisions. So this is an answer that we are seeking. But like Fubao said, considering the number of patients affected and the lack of any disease-modifying drugs and the very dramatic significant changes that we're seeing. So going forward, we strongly believe that this is no longer about whether this program is going forward as allowed by the FDA or not. This is more about working with the FDA to see how the program is going forward. So it's not a question of if, but more like the question of how. And getting the RMAT, therefore, is important for us to have continued access to the FDA in a more interactive manner instead of finishing the trial and then go and see whether they approve or not, because this is really a territory, an uncharted territory that nobody has done before. And considering the difference that we have seen and the unmet demand, so we are very hopeful and think that this is more, about going through the process and find out how FDA wants us to run the trial. And therefore, I think what IQVIA has been supporting us or helping us to launch parallel programs in APAC and in China, that would be something very interesting as well, because we are able to tap on the existing progresses to jumpstart, run parallel track, regulatory track in APAC in China, and where the data provided that we have a global design, the data collected over in China can also be used as human data to support our EU and US trial and vice versa.
Cool. In the interest of time, I've received a few questions. Sorry, time for Q&A. Let's start with Dr. Ping and then Li T, right? Yeah, please.
Hi, Prof. Lee. Thanks for the information. And thanks, Dr. Prof. Roger, for all the updates. So I just have two very basic questions. So I assume that your gene therapy actually helps the cardiac myocytes to actually increase, and then that's how the heart muscles get better and reduce in heart failure. I also see that your endpoint is actually reduction in mortality due to I guess due to heart failure. So where are we measuring in your this gene therapy? Are we measuring a improvement in the heart failure condition, or are we measuring the mortality rate for heart failure?
Yeah, so we're measuring improvement in the function of the ventricle, uh, and specifically the wedge pressure, which gives you a direct view of what's happening to the loading of the left ventricle, especially the pressures within the left ventricle, diastolic pressures. Now, the mortality association is based on long-term studies of HFpEF, which have measured wedge pressure and have shown how wedge pressure in the HFpEF population directly correlates with mortality. So if your wedge pressure at rest or at exercise are elevated above a certain degree, then your mortality is anywhere between 30 to 80% higher. So based on those changes that we see in our wedge pressure, we can correlate what the mortality will be in our HFpEF patients.
Okay, thank you. So one last question. So this is just a while short. We assume all these are chronic disease causing some of the heart failure or ischemic heart failure, things like this. Just wondering, will this gene therapy, when they improve the heart function, also improve the other chronic diseases like the hypercholesterolemia and various things like this?
Yeah, so if you look at, for example, at pulmonary and kidney disease, where you see most of the comorbid illnesses, so
Yes.
an increase in flow, a decrease in congestion will improve kidney function. Similarly for pulmonary function. So we believe that yes, there are some direct effects of some organs distal to the heart that will get improvement once the function of the heart improve. Indeed.
Okay. No, I'm good now. And thanks for the clinical information.
Actually, Doctor, I just
Sorry,
yeah,
can I just interrupt? Because I think a lot of the people on the call are not doctors and don't fully understand. And actually, in response to what Dr. Sui Ping just asked, actually, you don't increase the number of myocytes. So maybe if I can, sorry, Hajar and Ronald, I'm just going to interrupt to make it really simple for these folks to understand because most of them have no medical background. So typically, what we do most of the time now for patients with heart failure, now they are talking about two types of heart failure. One is preserved ejection fraction. That means the pump function is normal, but the heart is very stiff. So that also causes back pressure in the lungs and people can go in heart failure. That's the HFpEF they're talking about. All right. So you could see when you measure the ejection, EF is the amount of blood pushed out to the heart with every contraction. So that is HFpEF. That's what they're talking about. Then you have the part where the pump is not working well. That means it is functioning below what is expected to. So there's a one with impaired heart function. So two different groups. That's number one. The first thing you need to know is that for these kind of patients with this problem, they have a very high mortality. So for example, if you diagnose someone with heart failure, 10 years down the road, maybe for 100 patients, about 2/3 or 3/4 of them will perish. So that's the statistics you're looking at. So now what they are saying is that, okay, we have treatment options now. What are the treatment options? Drug options. So that's what they were explaining, the Entresto, the Forxiga, and diuretics, and etc. So that is the so-called the benchmark in which they are benchmarking their treatment is. So what is their treatment actually? So this SERCA, they are targeting a particular enzyme called SERCA2A.
Yes.
What does this do? Actually, this is involved in shifting of calcium from one part of the cell to another part of the cell. When this calcium moves, actually it causes the heart to contract. So it's very important for this function. So for many, many years, as you have seen in the slides they presented, many people have tried to, put the gene in through viral vectors to try to insert it to the cell to revitalize this enzyme, increase production of the enzyme so that this heart function can go back to where it was in the past. But as you are aware, the trial, so this is not new, this has been somewhere for a long time, but I think what they're showing you is now they have a better method of doing it. The science is correct. There's nothing, the science is exactly what is intended to do. And now I think what the Medera has done is they're able to do what the initial intention of what most doctors and scientists have been trying to do. How do we revitalize this pump function to improve? So in a nutshell, you have a spectrum of people, heart failure, very high mortality 10 years down the road, two-thirds to three-quarters die. You have the standard options with drugs. And now what they're trying to show you is that their therapy is superior to the drug therapy. Number two, this is research that's been done for a long time. They have a superior way of doing it. And then just to, Yong Peng, just to reiterate again, they don't actually increase the amount of mass.
Okay,
what actually is that they improve the efficacy and functioning of the mast cell. So I try to make this simple because I think most of the people on this call are not doctors. So they really don't fully understand a lot of this technical detail, but I'm just trying to make it very, very simple for everybody so that when you ask questions, you have a better sense of where we are, what questions to ask. And yeah, so I hope this helps. Yeah.
Yes. Thanks, Michael. Yeah, okay. So my misconception, I always thought gene therapy is about increasing more cells. So that's why I have this pre-concept. So thanks for updating.
Cool. I just want to leave the question for Li T, just to jump in. I'm sorry for the lack of time.
Thank Dr. Ronald and Dr. Roger for the explanation. Just want to draw comparison to the difference between Celadon and Medera, because a lot of these things, there's a lot of similarity, right? The Circle 2 IC, they're both used in Celadon and Medera. Of course, there's some changes in terms of dosage, in terms of the way IC, you talk about electric pump, and using a more efficient manufacturing process, capsid, right? So there are a lot of some changes, but it all has to do with increasing the delivery of the same underlying SICA2, right, to the heart. So the question is, what is the difference in terms of the clinical outcome you observe so far? Because early stage, the previous method also worked pretty well for Celadon, right? And we move on to phase B, and it turned out that the data isn't it didn't really work out in the later stage. So at this stage, we are still at a very early stage, 10 patients. And so from the clinical data, what is actually different? But how can we gain the confidence that this time is different?
Yeah, so I mean, if you look at the Celadon, I mean, I've lived through the Celadon data, and obviously I'm living through the Medera data right now. A lot of things, if you look at the Celadon data early on, in terms of ejection fraction, New York Heart Association, 6-minute walk test, all the parameters that we're seeing improvements in right now with Medera, we didn't see really improvements. Even the early data that were positive were based on clinical endpoints that were shown to be improving. But at the end of the day, as you can see here, EF was really not improved like we're seeing improvements in our HFrEF trial. Of course, at Medera, we didn't, I mean, at Celadon, we didn't do HFpEF. In terms of 6-minute walk test, we're seeing specific improvement with Medera, whereas we had a decrease in within Celadon. And most important, and also NT-proBNP increased at Celadon, whereas it decreased with us. But most importantly is the fact that we had such a low transduction efficiency. I mean, we're 0.5 to close to 100% now. So I think that is really a huge difference. But all the parameters that we basically compare to are improved compared to Celadon. So our confidence is much higher based on these data.
Yeah, I just want to, because I've been on a much longer call last 2 days ago where 45 minutes was spent on Celadon. I just want to point out that I got a feel that the two professors were so frustrated with the Celadon's progress, right? Also, their lack of technology available almost 15, 20 years ago. And then versus the clinical trial, the reason how it was set up, they forced it into phase 2B, right? Those are all reasons why they spent their own money to set up Medera because they didn't have that much say. They were scientific co-founders of the Celadon technology, even of UsBio, right? This time they're running the whole show themselves. And I wanted to point out, right, if you look back at the history of GLP-1, which is the hottest class of drugs today, the first version, right, is called liraglutide by Novo, and that only weight loss was 5%, right? Then it became a 16% one called semaglutide. And then the third generation or 2.5 generation is Lilly's tirzepatide. So it takes a few generations to perfect it. And it doesn't mean that it was a dead end at Lyra. In fact, I think that you can look at for this new Novo article where the chief scientist, right, was told to shut down the GLP-1 program at Novo before they struck gold. Yeah, so that's the background of it.
Yeah, understood. Do we have time for another question, or are we out of time?
Yeah, I think we're out of time, but please just ask the question and we see we have 1 minute
It's okay.
answer.
I'll send
Yeah,
it to you.
no.
I'll send it to you. Yeah.
No. Then, well, we will set up a separate call for Q&A. I'm sorry to put it this tight, but please just ask your question first, right? And then we'll see how that goes. Thank you.
You mean I asked the question now?
Yeah, just ask the question first. I'll see whether they have a short reply.
It's a separate question, as in because the last time round for Celadon is actually endorsed by many major pharmaceutical companies, right? Like I think it was Novartis, was it? Pfizer and J&J. So this time around, of course, we have IQVIA coming in, but no offense to David, right? But the IQVIA is a CRO, right? So not sure what's the difference. What kind of data are they seeing that gives them the confidence? Because I guess they're involved in the trial, but they're not really the domain expert. And so there should be two parts to this question. One is IQVIA, what are they seeing? And in terms of the conversation, is there any major pharma companies involved at this point in time?
Yeah, I can take this question. So things are happening by stages, and also we have learned a lot in the past 25 years about how best to sequence certain events. So IQVIA is coming in as our global CRO to synchronize our trials in the US, EU, and also China. And we're excited about working with them because of their capability of, being able to help us not only to complete the trials, to interact with the FDA in terms of our regulatory milestones, and also to help us commercialize when the time comes as well. And I think things are coming up very quickly. We're aiming to submit our BLA, the first BLA in 2027, 2028, as we complete the pivotal trials. So, and at the same time, they are now in the process of helping us to explore BD opportunities based on the momentum that we have already built. So we've been talking to several. We've been actually after we presented the late clinical trial at the American Heart Association just in November. We presented only half of the data, just the no dose cohort. We've been approached by several pharmaceutical companies, including some of the usual suspects like Lilly, J&J. We've been talking to Bayer, Astellas, Insmed, and quite interestingly, we've been approached by several Asian and then primarily Japanese and Korean companies as well. So, and also in China quite recently, without saying who specifically. So I think the reason why we are completing this round is that with the total amount of $80 million that we are raising for this crossover round, we'll be able to completely basically de-risk the program because will be the quote that we are getting from IQVIA Global US is that to complete the pivotal trial is about $40 million or so. So in that case, we think that by investing a relatively small amount for the kind of addressable market that we're looking at and the impact, the unmet need, and also the time it takes to get additional data, we'll be able to create a lot more value for the company and therefore our shareholders. So we're not talking about a typical cardiovascular trial of several thousand patients, and which is going to take years. So, but in the process, so the idea is that we proceed with the trial, $40-50 million to complete and take us to the next level, directly working with our sophisticated or industry investor, IQVIA, which is also going to be helping us with our global trials. And in the process, so 2026, we are already engaging potential BD partners, so as to create more value. So that's the approach, and very different from what you were saying about 20, 15 years ago, patient selection, endpoint, and all those are very different, I would say. I would say, in technology is common, right? And at least this is me and Roger. So we look at what can be done now, and history is just a lesson for us to go back and see if there's anything to learn. And also in this case, as Dr. Lim mentioned, I think technologically, the manufacturing method, for example, and also everything that goes with it's very different. So in a way, you just look at the cell phone that you have now 25 years ago with the first generation Motorola, that would be hard to imagine that, you can have access to the internet and do all sort of things like. So I think we're moving on. We're able to benefit a lot. The science is the same, but we're able to benefit a lot by the advances in all the other technologies that go with this.
Okay, I'll just wrap it up by saying that Ronald is based in Hong Kong most of the time, right? Because of he's working with IQVIA and us. In fact, he's supposed to be back in Shanghai for that final Mega Fund meeting in the end of February, right? So we're a short flight away. Please come in and see the Noble Heart technology yourself in their Quintong labs, right? I think a lot of investors would do that, especially if you are coming in this round. Yeah, so my apologies, we are running out of time. The other party's been waiting 20 minutes. We'll just drop off here, but I just want to point out that I'm going to put Ronald into our WhatsApp group so you can just follow up with the Q&A directly. Okay? Great. Thank you so much. I want to wish all of you a great New Year ahead, right? 马年快乐, huh? Okay?
Thank
Take care,
you.
guys.
Thank you.
Thank you. Peace. Bye-bye.
Thank you. Thank you.